Going to pick up where we left off the other day. So the other day we had talked about the kind of structure of skeletal muscles. I wrote up that flow chart that I had up there the other day. At the top you have the whole muscle made up of bundles of fibers and what we call fascicles. Remember, fibers are just a fancy way to describe cells. And what we got pictured on this page is a singular myofibril, which is made up of sarcomeres. Okay, sarcomere is a functional unit of a myofibril made up of various filaments, thick and thin, as well as the chitin filament that we'll talk about. And what we're going to discuss today is the relationship between the thick and thin filaments and how that results in muscle contraction. Before we do that, I want to talk about what proteins make up our myofibrils. Include myosin and actin. Myosin is the motor protein that's found in all three types of muscle fibers. Motor proteins are proteins that push or pull various cellular structures to achieve movement by converting chemical energy into chemical energy in ATP, the chemical energy of motion. Myosin is the only protein that makes up the thick filament. About 300 myosin molecules constitute the thick filament. Okay, we'll see a figure of it here in a couple slides. Okay. The major component of the thin filament is actin. Okay. Those two relate to one another in the fact that that's how muscles contract when myosin heads pull on the actin molecules. Okay. We'll see a little bit more about this in just a second. I'm going to go ahead and let you guys write out the stuff here on regulatory proteins and structural proteins though. I just said that myosin was the only protein that's part of the thick filament. Actin is the major protein of the thin, but you also have tropomyosin and troponin, which are classified as regulatory proteins, helping switch the contraction process on and off. And they do so in response to chemical release, chemical being calcium. Okay. The third type of proteins are structural, providing supports, helping to provide with proper alignment. They also give that elasticity and extensibility to our skeletal muscles. Remember, elasticity is that recoil after being stretched or contracted. Extensibility is the ability to stretch. Okay. The major protein that does that is titin. Okay. If you go back to that slide, that figure a couple slides ago, you get to see titin filament was listed. It's the third most plentiful. It actually gets its name for its size. That's what titin actually means. It stabilizes the position of myosin by spanning all the way from the Z-disc to the middle at the M line of our sarcomere. This is where elasticity and extensibility comes from. Okay. You'll want to know that titin does that. Okay. Dystrophin is a protein that links the thin filaments and sarcomere to integral membrane proteins in the sarcolemma. If you've ever heard of muscular dystrophy, one of the forms that causes this is due to defective muscular dystrophin. Okay, it's named muscular dystrophy. Okay. And then nebulin is going to wrap around the entire length of each thin filament, helping to anchor the thin filaments to the C-discs. But the main thing that we're going to talk about are actin or myosin, and then to a lesser extent troponin and tropomyosin, which we can see here on this next page. Now, unfortunately, I made copies before I found this figure that's on the left side of the page. And so yours is not nearly as fancy as the one that I have shown here. Essentially just a simplified version of it though. Okay. So, I said that proteins that make up the thick filament are strictly myosin molecules. About 300 of them organized in opposite direction of one another. One goes one way towards the Z-disc on one side, the other goes to the Z-disc on the other side. Okay. Each myosin molecule looks like you took two golf clubs and twisted their shafts around each other, and then the heads point off And the heads are like my wrist, or your wrist, any wrist that has the ability to go from an extended position to a flex position. Okay? They do so at this kind of hinge that we show here. Okay? The heads are the most important part. At the tip of the heads are these actin binding sites. That's where it binds with, you guessed it, actin. Okay? Look down here at each of these little beads. They have a myosin binding site. That's where the two meet to form what we call a cross bridge. Okay? They also have an ATP binding site, which allows them to become energized, because that's what ATP is. It's energy. Okay? That's what allows it to change its shape, to go from extended to flex, and flex to extended, to hinge back and forth, basically. Okay? Now, what that does is allows for it to grab a hold of the thin filament, okay, which is primarily made up of the protein actin. Now, actin is made up of these little beads called G-actins, arranged into two strands that are organized into a helix. That strand is called an F-actin, so we have two F-actin strands that are part of the thin filament. You don't really need to worry about any of that. Okay. You do need to know that they have the myosin binding sites, and in a resting position, they're covered up by one of our two regulatory proteins. We call those rope-like proteins, tropomyosin. Notice down here that those binding sites are covered. That tells me that this is a resting thin filament. Okay. When it is ready to contract, calcium binds to this set of proteins called troponin. And that causes the movement of tropomyosin. And we'll talk about this today. We'll have to show a video that illustrates this as well. Okay. So that's how we turn on and off the contraction cycle. Okay. And we'll talk about that today. Now, over here on the right, it should be the exact same figure that's in your lecture, showing you what happens to a couple sarcomeres when we go from relaxed to partially contracted and into maximally contracted. Okay, they've labeled out here the H zone, the I band, and the A band. Remember, the H zone represents the point in the sarcomere where there's only thick filament. There is no thin. I band is the opposite, only thin. Okay. The A band just represents the length of the thick filament from one side to the other. This will not change, because remember, the filaments do not shorten. What happens is the zone of overlap grows, and in doing so, the I band and H zone will shrink. So look what happens when we go from relaxed to contracted. Our green H zone gets smaller, doesn't it? Same thing for our blue I band. That's because our zone of overlap, which is this white region, has gotten bigger. Has the A band changed? No. Okay. It doesn't even change when we go to maximally contracted. In a maximally contracted, we no longer even have an H zone or an I band. All we have is basically zone of overlap. That overlap that exists between the thick and thin filaments. Make sense? So now that we understand structure, we get to learn how it all works. It all traces back to a nerve. Okay. Nerve signal coming from a somatic motor neuron. Somatic tells you this is the division that is involved with voluntary control. And motor tells you the signal is leaving the spinal cord going outwards. Okay. Or it could be from cranial nerve, so it could be coming from the brain. Okay. When that nerve signal arrives at our cleft, or at our knob, or bulb, whatever you want to call it. And it's the same thing that we've talked about before. And the reason that I talk about nerve signals and action potentials and synaptic transmission before I talk about this, because it should be right there in your minds, hopefully, somewhere. That action potential arrives at the knob, calcium rushes in through voltage-gated calcium channels, synaptic vesicles move towards the presynaptic membrane, released by exocytosis into the synaptic cleft. Same thing that we've seen before. Okay? We just really simplified it. All we're saying is action potential motor neuron releases acetylcholine. ACH is our neurotransmitter release. That's what we call the synaptic, or the motor end plate. Okay, this invaginated part of our muscle membrane is called the motor end plate, and it is full of these receptors called acetylcholine receptors. They bind with acetylcholine, or acetylcholine binds to it. Okay. The reason it's invaginated, meaning that it has these depressions, it allows for a lot of receptors. 30 to 40 million of them. There's more surface area, which means there's more membrane for those receptors. So that means more acetylcholine can bind to the receptors. And what those receptors do is when acetylcholine binds to it, it opens them up, which allows sodium to rush in, and to a lesser extent, not nearly as important, but potassium to rush out. What we're concerned with is the influx of sodium. And if sodium comes in, what happens to our overall charge inside of our muscle cell? Just like we saw earlier in the semester, sodium has a positive charge. We become, or let's say less negative. Okay. We're going to approach threshold. There's that word I was looking for. First class didn't get that, so good job. Was that you, Wyatt? Yeah. Great job, son. So, acetylcholine binds, sodium rushes in. If enough sodium rushes in, threshold. Okay. That means we've created a action potential in our muscle fiber. And just like in neurons, once we open the threshold, or once we hit threshold, open those floodgates, that action potential is going to spread like wildfire. Okay. This time, the signal is going to travel throughout the sarcolemma, not down the axon, because we're talking about muscle fiber. Remember, the sarcolemma is a fancy way to describe the plasma membrane, which is on the outside of our muscle fiber. We also learned last week that there were those extensions called T-tubes. They carry the signal inwards. Inside of the muscle fiber is what we're going to see here in just a second. Okay. So what we're describing is muscle fiber excitation. We're creating an action potential in our muscle fiber from our original action potential in the nerve fiber, or in our neuron, if you want to think of it that way. And this figure shows it off. Okay, acetylcholine binds, sodium rushes in. If enough of a strong enough local potential is created, we create that action potential, which is due to the flow of sodium ions in through voltage-gated sodium channels. That action potential spreads throughout the muscle membrane and then heads inward by way of the T-tubes. Okay, which we'll see in just a second. Now, those figures that I've shown here on the kind of previous couple pages are all from a different textbook. I kind of like them a little bit better because it kind of gives you step by step. And there's just a couple figures in your textbook. This is one of them. Kind of just is a more overall kind of summary style figure. Okay, I do like this figure because it kind of shows you that here we have our synaptic end bulb. Here's our muscle fiber. That point where they meet is called the neuromuscular junction. We'll talk more about that on Wednesday. Okay. Axon terminal receives that nerve impulse. Calcium's coming in. Acetylcholine is released. Acetylcholine crosses. The colectin binds to receptors. Sodium rushes in. We've created an action potential. Okay. Action potential then spreads into the muscle fiber by way of T-tubes, which we learned T-tubes are short for transverse tubules, and they're listed on this page and this figure as T-tubules. So that's why I wrote them on this page as. That's how we get our signal from the outside inward. So in our Step 6, it shows with yellow arrows the spread of an action potential away from our motor endplate along the plasma membrane, the sarcolemma, and then inward by way of the T-tubes. Propagated inward, which stimulates the release of SR, or calcium from the SR. SR were those membranes full of calcium. SR was short for sarcoplasmic reticulum. Okay. Since the calcium concentration in our cytosol is very low, generally, there is a gradient that draws the calcium out from these calcium release channels into the cytoplasm, the sarcoplasm. Okay. Now, these terminal cisterns are just the parts of our SR that are right next to our T-tubes. Remember, we call this 3 structures a triad. 2 terminal cisterns and the T-tube on the inside. Okay. So that calcium is released from these things called calcium release channels, which I'm going to show you a YouTube clip here in just a little bit. They're going to give it a more fancy name. You just need to know they are calcium release channels, which tells you exactly what they do. Release calcium. That calcium is very important because it's instrumental in the muscle contraction process. Without it, we can't have the formation of a cross bridge, therefore cannot have muscle contraction. And you'll see why. The events that lead from an action potential to the contraction is described with what we call the excitation-contraction coupling process, or just excitation-contraction coupling, which is a lot of times just abbreviated ECC. Okay. Excitation refers to the action potential aspect. Contraction, obviously, we're talking contraction process. Okay. So it begins with calcium outflow from the SR, binds to troponin, shifts to tropomyosin, exposing the binding sites, so it allows us to form what's called a cross bridge. And then we can go through what's called the contraction cycle, and that's what we're going to look at here in the next couple slides. So here is our thin filament at rest. Okay. The myosin binding sites are covered by tropomyosin, but we've bound calcium to troponin, which then allows step 9 to occur, and look at our arrows to show off tropomyosin is exposing our binding sites. Now, here we have it listed as active sites. Might not be a bad idea to put in parentheses, that's our myosin binding sites. Same thing. Can't remember active site, myosin binding site are the same thing. It'd be a good idea to write it down. Now this allows us to bind myosin to the actin. But we're not quite ready because right here, our myosin heads are not in a good position. They're already flexed. We want them to be extended, which is what we're showing down here with step 10, which requires ATP to be hydrolyzed or broken down to ADP MPI, inorganic phosphate. Because if my wrist is like this, can it flex anymore? No. But if I go like this, now it can, right? That's what we have to do before we form what's called a cross bridge, where that's basically just the connection that exists between actin and myosin, which then allows the myosin head to go into this position, what we call the power stroke, okay, which is one of our steps shown here on this page. As we perform the power stroke, the thin filament will slide across the thick filament, which is why we describe it as the sliding filament theory. And I've talked about this since last week. Muscular contraction, step in which the muscle fiber develops tension, they shorten. Model describing this is the sliding filament theory, stating that the actin and myosin myofilaments do not become shorter during contraction, which is why that A band didn't change its length a little bit ago. Instead, the actin slides over the myosin, which causes the whole muscle fiber to shorten. The power stroke is where we take that myosin head and we flex it. And in doing so, the thin filament is going to slide past the thick. Okay. Notice that for this to occur, we release ADP and PI. Okay. Now, what we have to do at this point, in order for a muscle to really shorten, because one power stroke, I think it only shortens a muscle like 1%. It's not very much, is it? Okay. When you go like this, flexing your arm, that's a lot more than 1%, isn't it? Okay. So what we have to do is break this, and that's one of our steps that's listed here on our cross bridge or contraction cycle. Now these 4 steps are all shown on the next page. And I will talk about them just as soon as you guys are done writing. Okay, so step 1 is ATP hydrolysis. We already talked about this. Taking our myosin head from its flex position to its cocked position, or extended position, whatever you want to call it, cocked, extended, same thing. Okay. In doing so, we have to use ATP and break it down or hydrolyze it to ADP and PI. And now our myosin heads are in correct position. Then they can form the cross bridge, like we just saw. Okay. Now this is only possible if those myosin binding sites on actin are exposed, and that only happens if we have calcium binding to troponin. Okay. Once we've formed a cross bridge, we can then go through the power stroke, or active stroke as we called it. We release ADP, and that allows the myosin heads to go like this, to flex. And in doing so, the thin filament slides across the thick. Okay. Now, in this position, we are in no shape to do it again. Because it's a cycle, this is going to continue to happen. So we have to utilize ATP again to break the cross bridge. Okay, which is step 4, cross bridge detachment. Now this is going to continue over and over and over again as long as we have 2 things. Calcium that's bound to troponin, causing a shift in tropomyosin, exposing the binding sites, and then we have adequate ATP concentrations. Now, when we die, we go through what's called rigor mortis. Okay. This occurs after death due to the release of calcium from the SR, causes this process to keep occurring until all the ATP is utilized, thus not allowing for cross-bridge detachment. So basically, we get stuck in step 3. So what does that do to our overall body and our muscles? They get stiff, right? That's why they call dead people stiffs. Okay? That's what causes rigor mortis. Okay? Our muscles get stuck in rigor. Okay? Any questions on any of that? Okay, so what we're going to do now is talk about muscle relaxation. The steps that lead to muscle returning to its resting position. Our original signal was an action potential from a neuron. So it makes sense that our original signal for relaxation is the cessation of a nerve signal from a neuron. So we stop releasing acetylcholine because we no longer have an action potential in the knob. Okay. And any residual acetylcholine is going to be removed by the enzyme acetylcholinesterase, or sometimes it's called cholinesterase, which breaks apart acetylcholine into acetate and choline. Now, in the figure that you'll find in your textbook, I'll show you in just a second, they actually show this as kind of step 3. This enzyme is going to be doing this even while our muscles are contracting. They just here show it as step 15. Okay, so we're always going to be utilizing acetylcholinesterase to remove acetylcholine, even if we are continuously releasing acetylcholine from our synaptic knob. Now, if we no longer have any acetylcholine binding to receptors, we can no longer have a local potential, which means no more action potential. Okay. So no more signal traveling down the SR and then inward by way of the T-tube. So our calcium release channels are closed. And now we open what we call calcium active transport pumps, or calcium ATPase pumps. Same thing. Now, 2 things are told to you when you see the words active transport. It requires what? ATP. Okay, illustrating the fact that ATP is not only needed for contraction, it's also needed for relaxation. And active transport means that we're pumping against its gradient from low to high now. Okay. So we're going to put it into SR, and there's also a protein called calsequestrin that helps us to store extra calcium back up in our SR. Basically, we're taking the calcium away from troponin, which is what we're showing here on this page. Calcium's removed from troponin. Look what happens to tropomyosin. It shifts back. Okay. So now that we don't have a cross bridge, we can cover back up those binding sites. Can we form a cross bridge now? No, because the binding sites are closed, or they're covered. Okay, and that's going to allow us to return to resting length, thanks in part due to Titan, which has that elastic component to it, allow it to snap back, but also the contraction of antagonistic muscles. Okay, like when I go from a flexed arm position to an extended arm position by contracting my triceps, that also helps to stretch out my biceps. Now, this is that figure that I was talking about, kind of that overall arching figure from your textbook. Yeah, they have it listed here as 9 steps. Starts with the nerve signal reaching the knob. Acetylcholine's release crosses the cleft, binds to receptors on the motor end plate, creates an action potential that spreads down our T-tubes, causes the release of calcium from calcium release channels. Calcium flows out into the sarcoplasm, binds to troponin, shift in tropomyosin, cross-bridge cycle occurs. All those steps involved in contraction, causing our sarcomere to go from an extended or longer position to a shorter position. Okay, we see the I band, the H zone disappear. What we're left with is a maximally contracted muscle. Basically, all it is cross-bridge or zone of overlap. Okay. Now, then we relax the muscle. We no longer have acetylcholine binding to receptors thanks to acetylcholinesterase. Breaking down acetylcholine, like I said, they use step 3, and continuously removing acetylcholine. That way, no more acetylcholine, once it's stopped being released from the motor neuron, will stop all this. Okay, calcium's pumped back into the SR, means it needs ATP through the calcium ATPase or calcium active transport pumps. No more binding, no more shift. Okay, allows the muscle to return to its original length. Now, I also gave you here 11 steps summarizing these events. Okay. You will need to know these steps. Now, am I going to ask you to list this 11 steps out on the test? No. But I might give you step 2 and ask you what happens next. Okay, or I might, say which of the following happens first and list off 4 things. Which of the following happens last. So I always tell students, you can walk yourself through these 11 steps by taking out a blank piece of paper and writing them out. You know your stuff. Now, I also, pretty good chance, you'll throw out figures. These figures here, I might put on the test and say, hey, what does this figure illustrate is happening? So you want to make sure you know what these figures are, what they're representing is happening. Sound good? This is awesome, right? Now think about just how amazing our body is. I've been using this same example all semester long. Hand on a hot stove, creates action potential in a sensory receptor that sends that signal upwards into our spinal cord. That release of a neurotransmitter occurs due to all those steps in a synapse, stimulates an interneuron, which then causes the release of neurotransmitters from that interneuron that stimulates the motor neuron, which creates an action potential that spreads out from our spinal cord. And then the release of acetylcholine at the neuromuscular junction causes all these steps to occur that cause our muscle to remove our hands from that hot stove. Think about all of that occurs in that split second. Not even a split second. We're talking milliseconds here. Pretty cool, huh? Hopefully you guys appreciate it. What I want to do now is show you some videos to help illustrate any of that. Okay. Hopefully those videos really help to illustrate what we just got done talking about. Okay. All right, moving on. Now we're going to talk about the importance of the number of cross bridges prior to contraction and the amount of force that can be produced, which is described with this thing called the length interrelationship. Okay. The length interrelationship, as it states here, for skeletal muscle indicates how the forcefulness of muscle contraction depends on the length of the sarcomeres within a muscle fiber before contraction. So basically, the length of the muscle before it contracts is going to have a direct impact on how much force that muscle can produce. And it's due to the number of cross bridges that can be formed. This morning I wrote over here, basically you can think of it like this. The more cross bridges you have, the more force can be produced, and force is the same thing as tension. So the length of the muscle affects cross bridge formation, which then affects force or tension production. And then I have 5 things listed here, which are illustrated on the next page. Now I've got 2 figures here on the next page. This one on the right is a little simpler. It's from your textbook. This one is the actual figure that's in the scientific journal article from when this was first discovered. Okay. This was first discovered in frogs. What they did to discover this was take a muscle out of a frog, hook it up to a machine that controlled how much stretch was placed on the muscle, and then they zapped it. And that zap created a twitch contraction, which is just a singular contraction. And they measured force as that twitch occurred. Okay. Now they put it at 5 different lengths. Point 1 is the shortest. Okay, that's the length of our sarcomere is almost 0. Okay. We show that down here. Our sarcomere is the shortest that it can go. Since it's the shortest it can go, it can't shorten any farther, right? Therefore, it can't produce any force. Okay. If we stretch it just a little bit, look what happens. We rise our force production way up. Okay. Illustrated in both figures. Okay. What that allows for is shortening to actually occur, and it has everything to do with the number of cross bridges that can be formed. Okay. Now we stretch it some more. Points 3 and 4 are a plateau part. Okay. Those represent the peak tension that we can produce. Okay. The maximal amount of cross bridges that can be formed, which maximizes the amount of force that we produce. Now, as we continue to stretch it, we descend until we reach point 5, which is where the myosin and actin no longer even overlap. Okay? If there's no overlap of the thick and thin, can there be a cross bridge? No. And just like I said, cross bridges affect force. So if there's no cross bridges, there's no force. Okay? Now, I gave you these 5 points. 1, 2, 3, 4, 5 represents on this figure 1, 2, 3, 4, 5. Okay? I might even put this figure on the test and I might ask you, what does point 5 tell you? Okay, what does point 3 tell you? Okay. I like this figure because it gives you kind of numbers to go along with it. This one's a little simplified. Okay. Now in green here, they kind of show you the optimal length. This is really not a super concern for us because our joints are built to maximize our number of cross bridges by affecting our length of muscle. Okay. We really will never ever get to a point where we're at more than likely 1 or 5. But this is something that does affect us, like when you're lifting weights or something like that. So it's all detailing back to the length at rest prior to contraction. So that's kind of cool. All right, last thing I want to talk to you guys about today is the production of ATP in our muscle fibers. We've talked today a lot in detail about the importance of ATP, not only for muscle contraction, but for muscle relaxation. Okay, to take our myosin head and extend it, to take it and flex it, to put ATP or to put calcium back in, thanks to ATP, along with some other things. We produce ATP in a muscle fiber 3 different ways. The first is through creatine phosphates, which is going to provide us with a little bit of energy for just a very short amount of time when we are maximally contracting our muscles. Notice I maximally put in italics. Okay. Now, what we do here is we take at rest ATP, which we have more than we need at rest, and we synthesize creatine phosphate, which is basically just a creatine molecule with a phosphate to it. That's what the CP stands for, creatine phosphate. So we take an ATP, that third phosphate, we form ADP because we've taken that phosphate and attached it to creatine phosphate. Okay. That's thanks to an enzyme called creatine kinase. That's super important. Okay. Now, creatine is an amino acid-like molecule, and when we go from relaxed to contracting, we can reverse that reaction, take that phosphate away, add it to ADP to form ATP. This process is anaerobic. We know that word by now means no oxygen is necessary for ATP to be formed. And like I said, this provides us with enough energy to contract maximally for about 10 to 15 seconds, which is like a 100-meter dash or a set of lifting weights. Now on Wednesday, we'll talk about why some people and what types of events people use creatine supplements in order to make their muscles stronger or have more energy. Okay. After that 15 seconds, we'll transition to the process called glycolysis, which is where we use glucose and break it down into what we call pyruvate, or pyruvic acid. Okay. This also does not require energy or require oxygen to form the energy, so this is anaerobic as well. It is a series of 10 steps, or 10 reactions, that doesn't really net us too much ATP at all. A whole net of just 2. We actually form 4, but we only net out 2 because we actually have to use a little energy to make that energy. Okay. We'll talk about this in greater detail later in the semester. Now, what we end up with at the end of this reaction is one of 2 things. We either form pyruvic acid, which then can be utilized in the next thing that we're going to talk about. That's going to occur if we have plentiful oxygen in the muscle. If there isn't, we'll convert it to lactic acid. Okay. Which lactic acid gets kind of a bad rap. I talk a lot about this in exercise physiology. A lot of people attribute it to things like muscle cramps. This morning somebody asked me when their joints pop, they've heard that lactic acid is involved in that. Absolutely not. Now, lactic acid is a known fatigue causer, but to produce lactic acid, you have to be exercising at a pretty high clip, okay, high intensity. I don't really get into that too much, but if you want to learn more about that, you can take exercise physiology with me next spring. This type of energy can provide us with activity for about 30 to 40 maximal seconds, maximal activity, 30 to 40 seconds of that. So, running, you're probably not going to finish a 400-meter race in that amount of time, but, you probably get most of it done anyway. And then the final way that we produce ATP, and it's the most efficient, is through aerobic cell respiration. Aerobic telling you that it requires oxygen. This occurs within the mitochondria. This is where we go through the Krebs cycle and then eventually the electron transport chain to produce a total of somewhere in the neighborhood of 36 ATP. And then releasing a lot of CO2 and water, as well as a bunch of heat. Okay. Now, if we're talking glucose, 36. If we're talking about fatty acid, talking upwards of about 100 ATP. This is going to be what we transition to the longer we go. Okay. Activities longer than 10 minutes, we're talking 90% or more from aerobic means. Okay. Marathon, pretty much 100%. Okay. And that actually is shown here in the figures that are on this page, or a singular figure. Okay, showing you time, 1 to 3 seconds, 100% anaerobic. By the time we reach 10 minutes, it's 90-10. 10 minutes, it's 80-20, almost 90-10 there by 30. And then, 120 minutes, 2 hours, it's completely aerobic. And then something that's kind of cool, comparing different types of events. Okay, so a lot of you guys are athletes out there, so you can kind of look to see what your event is. You don't need to know any of this. This is just all from my exercise phys textbook. That is science. I feel like it's kind of cool. Okay, any questions?